Hello, welcome back to Principles of Organismal Biology. Today we're going to learn about plants. Yes, more about plants and about how they grow. Here I brought one of the branches of my mulberry tree. This tree is growing vertically and horizontally. Here we have one little branch that has some meristems at the top. Hopefully you can see this leaf is coming in this side, and in here we have a meristematic region. This will allow the tree to keep growing, and this meristem is a primary meristem that helps with the growth going upwards. In the roots, we're going to have a similar meristem, a group of cells that are going to help the roots to grow down, right? Trees also can grow, as I said, horizontally. Not all plants can have that type of growth. That growth is called secondary growth. And here this is a stick. And you can see how that branch became this thick with time. And that is due to this ability of some plants, these trees, to form wood through secondary growth. Before jumping into today's class, I would like to remind you that we have the second midterm this Friday, in October 16th, and the exam is cumulative. Also remember to complete the pre-test or this quiz, which is also cumulative, and it's you're gonna get 20 points after completion. You will have 40 questions for your midterm and 50 minutes to answer that. And in your pre-test quiz, you're gonna have 20 questions and only one attempt, right? And 50 minutes to complete. So let's learn about plant growth and how plants grow. In here, you can see a fossil in the petrified forest in Arizona. So look at the landscape and describe it. So yes, this is a desert, and it is very unlikely to find these type of trees. You don't find these type of trees nowadays in the petrified forest. So how did this fossil arrive there? Well, actually, this place was dominated by gymnosperms in 235 to 200 million years ago. That means that the landscape in Arizona has changed dramatically and has aridified with time. Today, we're going to learn about the different organs that plants have and the type of growth that they can have, such as primary growth and secondary growth. For that, I would like to make this comparison. So in humans, we have cells. Let's look at this figure. We have a cell from the intestine, and this is particularly an epithelial cell. And then when we have a group of the same type of cells, we form tissues. And in this case, we have the epithelial tissue. And several tissues form the organ. In here we have the small intestine. And the organ system is going to be formed by several organs. So this is the digestive system. And several systems form the organism. In this case, they form us, right? So the same thing is going to happen in plants. We're going to have organs composed by different tissues and then tissues that are composed by cells. Look at this plant. We have the shoot system above ground and the root system below ground. Most plants have this type of organization, but there are some that are slightly different, and not all of the roots are below ground, but this is the common rule in plants. The root system is going to have one organ, and that's the roots. And the shoot system, the system that is above ground, is going to have two organs. The stem, right here, follow my mouse. This is the stem. And then we're going to have other type of organs called leaves. So we have several leaves in here. So the roots are going to rely on the sugars produced by leaves. So the leaves are the ones that produce or that are making the photosynthesis. That means that they are fixing the carbon into glucose. So these sugars need to be transported to the root system in order for the root system to grow. Now, the shoots rely on the roots because the shoots also need water and minerals in order to grow. So this is actually a bidirectional communication that happens in plants. Let's learn more about the root system. So their function is to anchor the plant, right? They create an anchor to support the whole plant. They are going to absorb minerals and water, and they are also, in some plants, they can store organic nutrients. We're going to have two types of root systems. And if you remember your class about angiosperms, we learned that we could identify two different types of groups in angiosperms. The monocots, which usually have fibrous roots, right? And the eudicots, which have taproot. In the case of the taproot, we are going to have a main root and then some secondary roots or finer roots coming out of this main root. This is a carrot, and you can see how the carrot keeps growing and growing, and the main root is the thing that we eat. And then if you are careful when you're eating your carrots, you can see the little eyes that are the secondary roots, or the points where secondary roots emerge. In the fibrous root system, what happens is that actually a main root is formed, but it dies, leaving behind only these fibrous, finer and non-very dominant roots, right? So in this case, in the taproots, we have one dominant root, and in the fibrous root, we have several, if you want to see it that way, or none. The main root is going to emerge from the seed. So this is a seed of a eudicot, and you can see the two cotyledonary leaves in here. And the root emerges as well, and rapidly is going to form these root hairs. Look at this image, and you can see hundreds of root hairs that are going to be able to absorb the water and minerals very quickly. These hairs are very, very fine, and they are extensions of the epidermal tissue. These are going to create an increase in the surface area, so they will absorb more water and minerals. In here, you can see different adaptations of the roots. In maize or corn, we have adventitious roots or prop roots that help stabilizing or with the stabilization of the stem. The storage roots are other type of modification that some plants have. In this case, look at the beets, but also the carrots. So in this case, these plants will store a starch. So a lot of sugar in these cells in the roots. And that helps the plant to go through winter and then use these sugars in the next year. Right here we have an example of mangroves. And these mangroves live in aquatic environments. And their roots are submerged in this water, salty water, right? They live by the sea. And they need to get oxygen in order to survive. The roots need oxygen. So in these conditions, what these trees have evolved is something called pneumatophores. And these are roots that emerge from the ground and above the water level to be able to capture the oxygen. So they are called air roots. The strangling roots are aerial roots, and you can see that in some monumental trees, such as the fig trees in Angkor Wat. And in this case, this tree is actually destroying this archaeological monument. So that has a conflict because the trees are very iconic, but also the archaeological site. So people are debating about what to do, if saving the archaeological site or saving these trees. Another example of adaptation of roots, you can find it in the buttress roots, which are actually very an extension on the base of the tree, but that's coming from the roots, and it really helps to support massive trees. So trees that are very tall, if they don't have this buttress, they will crack. But by having this extension or wider base, the trees can be very strong and support and grow very tall. This is just another set of pictures for you to see these mangroves and how the roots emerge in some cases, and how they create this microenvironment for some aquatic life to thrive there. Let's learn about the shoot system. The shoot system is the one that usually occurs above ground. And as I said, it's going to have the stem and the leaves. And the stem is going to be this organ that has alternating nodes. What's a node? Node is the connection, or is the place where the leaves attaches to the stem. So here, for example, we have one node right here in this side, and the next node will be in this side. What is the next node? So, well, the next node will be right here. So that's the node. And the internode is going to be this part, the length in between the nodes. Those are the internodes. Let's see if I can explain it with this. In here we have a node. This is the attachment of the leaf to the stem. Where is the next node? We have another one here. And this region will be the internode. Okay? What is the function of the stem? Well, think about this. Now we have a bunch of leaves, and without the stem, all of the leaves will be blocking each other. And leaves, we mentioned before, they perform photosynthesis. So if they are blocking each other, they won't have access to light. So therefore, elongating the leaves and separating them will maximize the photosynthesis. Now, leaves are going to have, usually, they have two parts. One is the blade, right? Like here, this will be the blade. And this little stalk will be called petiole. So similar to this, I'm going to crunch this or pull it. Now, we have the blade, is all of these. And then this little part will be called the petiole. Leaves, if you look closely, they will have pores. We have mentioned this before. They are called stomata, and well, the stoma, and they are associated to guard cells. So here in this picture, you can see this pore open, the stoma, and the guard cells around. This pore is open because plants need to exchange gases. So I say that plants photosynthesize, and for that, they need to have access to CO2, and they take the carbon from the CO2 and fix it into some carbohydrate molecules. But the byproduct will be the liberation of O2, the oxygen. But in the process, the plant, if it has the pores open, it will lose water, right? The water that was coming from the soil that was acquired by the roots, it will get loose in form of a gas by the evapotranspiration that plants have. So plants need to regulate the aperture or closure of this stomata. And they do it also regulating how much photosynthesis and how much water is available, or how much photosynthesis they need to perform. Now, there are also adaptations of the stem, and some plants have developed rhizomes. So in this case, we have a plant, and you can see the rhizome is actually a modified stem. It looks like a root, a very thick root, but it's not a root. That's an underground stem, and from it, the roots can emerge. So these are real roots. But this part is the rhizome, and that's a stem. Another modification of the stem is called the estolon. And the estolon helps to propagate a plant. So in this case, we have a strawberry, and this strawberry, it's cloning itself and forming a new plant. So they create this estolon, these appendages that they will, when they land at the extreme or the opposite side, they will touch the soil, and then from it, they will start developing different leaves and a root system. So with time, you will form several plants that are identical to the mom. Another modification are tubers. So tubers are also stems. And one example that we eat and that we love are potatoes. So potatoes are modified stems. And the eyes that you could see right here, for example, are the places where the roots will emerge. So in potatoes, it's interesting because potatoes have estolons and also these modified tubers. And tubers are gonna store a lot of starch. So similar as what we saw before in the roots that can storage carbohydrates, such as carrots or beets. In here we have stems that store a lot of starch. I have a question for you. So this strawberry, the scientific name is Fragaria vesca. It has stolons, which form new plants, as we have described before. It has 7 chromosomes in haploid condition. Therefore, a hexaploid plant will have 42 chromosomes. What will be the ploidy of a new hexaploid plant originated from the stolon? A, 21, B, 14, C, triploid, D, 7, E, hexaploid. Okay, the right answer is hexaploid. That's the ploidy, right? I hope that was easy. Let's learn about the 2 types of leaves that we can find. We have more types, but these are the most common types that we find. A single or undivided blade. This is a type of, it's an example. You can find it in oaks. You will have one leaf, and you have the petiole here and the blade. It's all of this. And please pay attention to the position of the axillary bud. Now, let's compare to the compound leaf. In this, we're gonna have multiple leaflets. So this means that all of this is one leaf. This is the petiole, and these little guys that look like leaves are actually leaflets. And then we describe this as a compound leaf, and that what it helps is to find the axillary bud. So in here we have this axillary bud that is telling us that all of this is a leaf, that it got modified through the evolution. So let's take a look to this, and let's find, hopefully, we can put this in focus. And you find this leaf, and then you find the axillary bud. And the axillary bud is right here. And different plants have evolved to have different forms, and that's depending on the environment that they have. For example, some of the pine needles that we have covered before, they actually look like needles. The pine leaves look like needles. In the case of junipers, which is another gymnosperm, we have scale-like leaves. So they are very short. And in these 2 cases, these plants have evolved to have these reduced leaves to overcome the lack of water. But look at all of these different forms of leaves. So this is another organ, right, that plants can have. In here, we can see spines that are coming from a prickly pear or nopal, and the spines are actually leaves, modified leaves that get really, really hardened with time. Tendrils in pea plants are modified leaves that have a function to climb. So these plants need to reach up because they need to photosynthesize. So they use these tendrils to grab around any surface, usually plants around, and they grab and twist around and then pull. And therefore they use that to have more access to light. Other plants form these reproductive leaves. So the whole plant can be producing these tiny leaves, and each of these leaves have or is able to produce a new plant. So you can take a lot of succulents have this property. So you just grab some of these leaves and they will regrow a new plant. But there are some that even on the maternal plant, they will start emerging some roots. That's very cool. And you can propagate them very easily. Another type of leaves are the storage leaves. So yes, we have covered that the stems can be used to store sugars or starch. There are roots that can be used to store nutrients, but also leaves. And one example is the onion. So look at the onion, you are very familiar with, I hope. And if you peel the onion, what you're peeling are a set of leaves. And these leaves are covering the stem. So yes, we also have the stem in the onion, but the stem is very, very reduced. And at the base, you will have these roots, all of these roots. Now, this is a magnificent example of how plants have evolved different modifications to adapt to different environments. All of these are carnivorous plants, and all of them have evolved to grow on soils that are depleted. So therefore, they found a way to deal with it and acquire some of these nutrients by eating or absorbing the nutrients from insects. So all of these plants, like the Venus flytrap, the Drosera, and these pitcher plants, they are distantly related, but they have evolved a similar mechanism. Watch this video if you want to have fun. I'm going to be posting it in Canvas for you. It's optional, but I have a lot of fun watching it, so hopefully you enjoy it. And if you're not the silly type, just skip that. But I think it's a good video for you to enjoy this class as well. So I have a question for you. Distantly related plants acquire their ability to eat meat through similar genetic changes. Plants typically produce enzymes that break down a polymer called chitin as a defense against fungi, right? Because fungi like to eat plants, so the plants really have to evolve to defend themselves against fungi. Carnivorous plants have repurposed the enzyme to digest insect exoskeletons, which are also made of chitin. This is an example of A, mutualism, B, convergent evolution, C, monophyly, D, horizontal gene transfer, E, the perfect Halloween costume for a botanist. Okay, so this is an example of convergent evolution. So and the secret on this, to answer it right, is that they are distantly related plants, right? So distantly related plants that evolve a similar mechanism to digest these insects and absorb the nutrients from them, but they are distantly related. So that's convergent evolution. Now, how do root and shoot systems grow? The answer is through meristems, right? But what's a meristem? So they are a group of cells that are going to keep dividing and dividing. So the meristems are perpetually embryonic tissues and allow for indeterminate growth. That means that plants always grow and grow and grow. It is most likely that plants die for an infection or because if they grow and grow and grow, then they don't have enough support, but they have the ability to keep growing and growing and growing. This is equivalent, the meristems in plants are equivalent to the stem cells. So they are a group of indifferentiated cells that we have. For example, we have when we're babies, but we also have some stem cells in certain part of our body, like in at the tips of our nails. We have some stem cells, such as that if we injure our finger, it can regrow under certain conditions. Please don't do that. Don't try to cut your finger to test that I'm wrong, but that's under certain conditions, people have been able to regrow their fingertips. And there is a lot of studies about it, and one of our colleagues in the biology department, Dr. Ricardo Sayas, studies planarian to see how they regrow their the body. So if you are interested in this topic, I suggest you to contact Dr. Sayas. If this is how it will look if humans will have indeterminate growth. But that's just silly, we don't have that. So we have two types of meristems in plants, the apical meristem and the lateral meristems is the second. The apical meristem is going to be located at the tips of both the roots and the shoots and the axillary buds in the shoots. They are going to be responsible for the elongation of the shoots and roots. And this is what we call the primary growth, right? And it's going to be growing in this direction, vertically, right? Whereas the lateral meristem is going to increase in thickness, and that is going to be called the secondary growth. But not all plants have secondary growth, and therefore not all plants have lateral meristems. Only woody plants have this type of growth, secondary growth. This is a figure for you to see the primary body. So the primary growth, and here we have the meristems, the apical meristems of the shoots, right here, and the axillary buds. And then we have at the tips of the root, we're gonna have this meristem. So this primary meristem of the roots. So that's gonna help, as I say, to grow vertically. We also have, if we make a cross-section of this shoot, we're gonna find 3 different tissues. So this is in blue, and the outside we have the epidermal tissue, and that's very similar, right? If we look at our skin, at the top we'll have our epidermis. So plants have their epidermal tissue. Then we have the vascular tissue, and you're familiar, you have heard the word, the vascular tissue, and we say that some plants or vascular plants have vascular tissue, and for example, bryophytes do not have vascular tissue. So some plants have this vascular tissue that is represented here by these ovals, and we are going to have phloem and xylem forming this vascular tissue. Everything in yellow is part of the ground tissue, and in here it's called pith or cortex. Everything that is towards inside is called the pith, and everything that is towards the outside is called the cortex in the shoot. So roots are going to have also these 3 different type of tissues that I explained. And in here you can see that. So this is a close-up of a root of an onion. So look at this. We're zooming into the tip of the root, and if you look at the actual cross-section, a real cross-section, you will see this organization. Hopefully you can see this division. Look at here, and these cells look different than these cells. These cells in the outside and the tip only are called the root cap. And this root cap is covering these meristematic cells. Remember, these meristematic cells are undifferentiated cells that are dividing constantly through mitosis. And they divide and divide. So these are very precious, important cells that plants want to protect. The root cap is offering that protection. Now imagine if you were swimming in a swimming pool full of rocks, that would be very painful, right? So imagine these guys, these plants actually are going through a lot of rocks. In the roots, these very soft cells, baby cells, need to be protected. So the root cap has this function, as I mentioned. Here in orange, we have the apical meristem, and this one is the one that is going to be dividing to differentiate later into these three different type of tissues. So in the outside, we're going to have this epidermal tissue in blue. Then we're going to have the ground tissue, and in here it's called the cortex. As I said, if the ground tissue is in the outside of the vascular cylinder, we are going to call it cortex. And then the vascular cylinder is pretty much our vascular system. So all of these cells differentiate into these three types of tissues. Look at the root hairs. Hopefully you remember that at the beginning of this class, I showed a picture of an emerging root developing a tons of root hairs, and I told you that those are epidermal cells. That means each hair is one cell that really elongates. Imagine you need to extract DNA from this plant, from this onion plant, for your research, because hopefully by now you have been accepted into a lab so you can gain some research experience. What region will have more DNA in 1/16 of an inch from this fruit? A, like in vascular cylinder. B, these root hairs. C, the vascular cylinder again, but more towards the base or the tip. D, the cortex. Or E, the region of the meristematic cells. Well, the right answer is E, because as you move, as hopefully you can see here, that these cells are dividing and they are very small, but as they divide, they elongate. So this is the zone of cell division, and then as they develop and they keep growing, they elongate, and later they differentiate into these three types of tissues. So the answer is E. Now, if we look at the shoots and the apical meristem of the shoot, we're going to have this organization. At the top, we're going to have the apical meristem, and surrounding this, we're going to have leaf primordia. So the apical meristem of the shoot doesn't have the root cap, right, or similar structure, but it does have leaf primordia that keep protecting this apical meristem of the shoot. These leaves, with time, these are baby leaves that as time passes and this primordium or these meristems keeps growing, elongating, and let's say now it's up here, these leaves will become these larger leaves that you could start seeing in here. And with time, these axillary buds will also form, and they will have these other meristemic or meristematic cells. It's very important to know that these axillary buds are going to be very important because they are dormant cells. And from these axillary buds, we can have new branches that can emerge, or for example, flowers can emerge from these axillary buds. Now the lateral meristem, it's going to form secondary growth, or it's going to have secondary growth. And we're going to have two different types of meristems in plants that form wood. And the first one is the vascular cambium. And this vascular cambium is going to add layers of vascular tissue that is called as xylem and phloem. So the vascular cambium adds xylem and phloem. You're familiar with that. But now we're going to have also the core cambium. And the core cambium is going to be a meristem that is going to be replacing the epidermis. And it changes from this soft epidermis that I was showing you before to something that is called the periderm. And that includes the cork. So if you look at one tree, you will see in the outside we'll have the bark, right? And that is part of this periderm. And this is very thick and tough. So it replaces, the periderm replaces the epidermis, which is softer and very delicate, or more delicate. So secondary growth doesn't happen in all plants. Look at this bean plant and tell me, do bean plants have secondary growth? Well, no, right? So they don't have this solid trunk, or they're not very woody. So not all plants have secondary growth. This is a representation of how the vascular cambium, how the vascular cambium works. In orange, we have the vascular cambium. And this vascular cambium is going to have initials, or cells called initials, that are going to be dividing. And the division of this vascular cambium is going to be producing phloem towards the outside and xylem towards the inside. Remember, I told you that in angiosperms, monocots have scattered vascular tissue. However, eudicots have the vascular system arranged in a ring. This is key because some eudicots that have the vascular system arranged in this ring can form the secondary growth because of that property of having the vascular system arranged in a ring. So here is this ring, and as I say, they divide, they form phloem outside and xylem towards the inside. But if you might be wondering, if they divide, then how they retain the vascular cambium, the cells that form the vascular cambium? Well, they alternate. So this is an initial of the vascular cambium, and then this cell divides in two cells, right? Mitotic division is going to always form two cells. Well, then the cell on the top, the blue cell with an X, forms or differentiates into the xylem, whereas this one in red remains as the initial, or the vascular cambium type of cell. Now, in the next round of mitotic divisions, this cell is going to divide, but now the phloem is going to get formed towards the outside, and this other cell remains as the initial. And then again, this cell divides again and forms more xylem, and then it alternates and forms phloem. Well, that's the way that this happens. So the one cell remains as the vascular cambium, and the other one either way forms the phloem if it's facing the outside, or more xylem if it's facing the inside of the tree. Now let's take a look to this figure and see what it happens when we have a baby tree. So this is a stem that is growing, or it could be a branch. But imagine this is a baby tree that it's just growing for a couple of years. And this is this year's growth. So in this year, if we make a cross section, we'll see this organization. We'll see the pith and the cortex, which are the ground tissue, and the epidermal tissue in the outside. And we have the primary xylem and the primary phloem towards the outside. Primary xylem in blue and primary phloem in brown. And look at how these vascular bundles are organized in a ring. If we move in this direction, we can see older growth. So this part of the tree is older, and this at the bottom is even older. If we cut now this section in here, if we cut the tree around this area, we can see that now this tree has accumulated a little bit of secondary xylem towards the inside and secondary phloem towards the outside. And see how the primary phloem and primary xylem are moving apart as these secondary xylem and secondary phloem forms. And in red, what do we have in red here? So this is the vascular cambium. And as we move in time in this tree, we can see that the xylem, more secondary xylem and more secondary phloem gets accumulated. So this is another year of growth. But what happens also in the periderm? So I say that the periderm is going to replace the epidermis. The epidermis was formed in the first year, and we're going to have this cork cambium in dark blue formed in here. And with that, we're going to be forming this cork in the outside, or yeah, the cork and the bark. When you peel a tree, if you have ever peeled a tree or if you've seen that, the bark is actually this periderm plus the phloem. The phloem also peels. That hopefully is telling you that the phloem actually is thin compared to the xylem. That means that more cells differentiate into the xylem during these mitotic divisions that we have. And this looks like that. So a maple, if we cut a section of a maple, of this trunk, will have true secondary growth, and each of these rings will be 1 year's growth. Whereas if we look into a palm, remember a palm is a monocot. So what do we have in monocots? Do we have secondary growth? Yes or no? Well, we don't have secondary growth, right? Because the vascular bundles are organized, scattered through the stem. So they form a type of a thick trunk that provides support to palms, but that's not considered true wood, and that's not considered through secondary growth. Secondary growth is only the result of the presence of the vascular cambium and the cork cambium, and palms do not have that, right? They don't have rings or vascular bundles arranged in rings. What palms have actually are parenchymatic cells. All of these are parenchymatic cells, and the vascular bundles are contained around these parenchymatic cells. Secondary growth is characteristic of gymnosperms and woody eudicots, but not monocots. As I was saying, you could look at the rings in a tree and determine the age of this plant. For example, in here we have this trunk, and if we cut it, we'll see the following. Here we have this xylem. This secondary xylem that you could see. You can count the amount of wood that was produced each year. And in the outside, we'll have the secondary phloem. And as I mentioned, the bark contains the phloem plus the cork and the cork cambium. And the periderm is going to have the cork cambium and the cork. I want to ask you this question. Where is the water, nitrogen, and phosphorus being transported? And where are the photosynthetates being transported? So the water and nitrogen, phosphorus, and other minerals are being transported through the xylem. So right here, all of these xylem is being used to transport the water and some minerals. In contrast, the photosynthetates are being transported through the phloem. So right here, this secondary phloem, it's being used to transport the photosynthetates. This is how a trunk or a piece of wood will look when you cut a tree, and a conifer, for example. You will have in the outside this bark, right? This is really tough. And in some trees, the bark forms very different type of, or different trees have different types of bark. And some of these features allow us to differentiate between different types of trees. You can also see the secondary xylem in here, and the hardwood, that's the youngest wood, compared to the subwood, which is older wood. And then we will have the vascular cambium and the secondary phloem plus the epidermis. Some trees can live very long times. I think I've mentioned before that Pinus longeva can live thousands of years, 5,000. I think there are some trees that have been recorded to live longer than that. In here, you can see a picture of a cartoon that shows the first year growth right here. And all of these rings are different years of growth. And that is because during the rainy season, the xylem tends to grow thicker. So during the rainy season in here in California, we have it in winter. So the trees will usually form a thicker xylem during that season. But during the dry season, the xylem will be very, very thin. And that is the difference that we see in these rings. So that means that if we look at a brown and a dark color of these rings, that will add up to a year. So 1 year growth will have the light brown and the dark brown. In here, we also have a scar from the forest fire that was registered during the formation or during the growth of this tree. This particular picture is a real picture of a cross-section of a tree, and we could see that the thickness of the rings were really thick in these years. So this tree was born in 1527, right, in the 16th century. And you can see that during the 16th century, we have an abundant amount of water because the rings are very thick, but suddenly we have a massive drought. It is called the mega drought. And this dendrochronology, this is the study of the rings in trees, can tell us how the climate was in previous years or in previous centuries. And if we plot this and this data, we can see the thickness or the ring width index and the years. So the mega drought will be right here, and we can compare it against other more recent dates that we have more abundant seasons. This is in the Mongolian tree rings, and this is very helpful to us. You are probably familiar with this tale. So this is the Jack and the Beanstalk. It's an English fairy tale that the author is unknown, but that was published in 1734. And I would like you to think about this and tell me what you think. So during the night, the magic beans cause a gigantic beanstalk to grow outside Jack's window. The next morning, Jack climbs the beanstalk to a land high in the sky. He finds an enormous castle and sneaks in. Okay, it is not real, but what could have helped with the rapid growth of the beans? A, high photosynthetic activity. B, high activity of the apical meristem. That means mitotic divisions. C, auxin production. D, lignin production. And E, all of the above. Yeah, we have covered that plants, in order to grow, they really need to do something, and that's photosynthesis. So photosynthesis, high activity of the photosynthetic, high levels of photosynthesis should help with this, as well as the rapid mitotic division, right? Because plants need to grow, and in order to grow, they have to undergo mitotic divisions. Lignin production, lignin will add support to the cells, right? You find this lignin in in the xylem and in other parts of the plant that provide extra support. So if you can think on this, that will add extra support to the growing beans. And the auxin production, we haven't covered it, but that's another type of, that's a type of phytoregulator or phytohormone, plant hormone, that produces elongation of the shoots and roots. So all of the above, it's the right answer. However, even though they are magic beans, this is impossible because of what? Because they don't have secondary growth. So remember, this is what I showed you before. The pea plants have these tendrils, and they don't really form secondary growth. So that will be really impossible in this for us to see anything like that. Well, that was a silly tale, but I hope that was informative for you to connect some points about plants, and I hope that you enjoyed this class. Thank you.